DNA is a double-stranded helix that stores the permanent genetic master blueprint for building and running your entire body. RNA is a single-stranded molecule that copies genetic instructions from DNA and carries them out to build necessary proteins. mRNA, messenger RNA, acts as the cellular messenger, taking genetic codes from DNA inside the nucleus directly to protein factory ribosomes. tRNA, transfer RNA, acts as a cellular delivery worker, grabbing specific amino acids and matching them to mRNA codes during protein assembly. Nuclear DNA, located inside the cell nucleus, nuclear DNA is inherited from both parents and determines your unique physical traits and features. Mitochondrial DNA, located in the cell's energy powerhouses, mitochondrial DNA is inherited exclusively from your mother and powers cell functions. Chromosomes. Chromosomes are tightly wound structures of DNA. Human cells typically contain 46 chromosomes in almost every cell nucleus. Genes. Genes are specific segments of DNA along a chromosome that contain precise instructions for making single proteins or specific physical traits. DNA replication. DNA replication is the process where a cell creates an exact duplicate copy of its entire DNA before dividing into new cells. RNA transcription. Transcription is the process where a cell reads a specific segment of DNA to create a temporary matching mRNA molecule. Translation. Translation happens at ribosomes, where the code on mRNA is read step by step to string together chains of amino acids. Protein folding. Once formed, amino acid chains fold into complex 3D shapes to become functional enzymes, structural fibers, or body hormones. Ribosomes. Ribosomes are tiny cellular molecular machines that read RNA sequences and synthesize thousands of proteins every single minute. Rough endoplasmic reticulum. The rough ER is a membrane network covered in ribosomes that helps fold, modify, and transport newly produced proteins across the cell. Telomeres. Telomeres are protective caps on the ends of chromosomes that naturally shorten every time a cell divides, acting as an aging clock. Epigenetics. Epigenetics studies how your diet, stress, and environment can turn specific genes on or off without altering the underlying DNA sequence. DNA mutations. Mutations are random typos or errors in the DNA base sequence that can cause genetic diseases or drive evolutionary changes over time. CRISPR gene. CRISPR is a revolutionary gene editing technology that allows scientists to find, cut, and edit specific DNA sequences in living cells. Exons. Exons are the active coding regions of a gene that contain the exact sequences needed to create real functional proteins. Introns. Introns are non-coding segments of RNA that are cut out and removed before the final protein synthesis takes place. Stem cells. Stem cells are master unspecialized cells that have the unique ability to transform into any type of specialized tissue in the body. Differentiated cells. Differentiated cells are specialized cells like muscle, brain, or skin cells that have locked into a single specific lifetime role. Apoptosis. Apoptosis is programmed cell suicide, where damaged or old cells safely destroy themselves to protect the surrounding healthy tissue. Cancer cells. Cancer cells suffer DNA damage that disables apoptosis, causing them to divide uncontrollably and form dangerous tumors. If this video cleared your confusion even by 1%, make sure to hit that subscribe button. Support, and let's keep decoding the world together. See you in the next one. Thanks for watching video.